Okay, so I am going to talk to you today more about Magell2 and about the work that we've been doing to try to understand more about the gene, the protein, and about Schaaf-Yang Syndrome. So the Magell2 gene was discovered really at the turn of the millennium as part of a search for genes whose disruption causes Prader-Willi syndrome. Many other genes for genetic disorders, like Fragile X syndrome, for example, cystic fibrosis, muscular dystrophy, were actually discovered longer ago than Magell2, so more in the late 80s and 90s. But genes for ultra-rare genetic disorders are still being discovered today. Over the last 20 years, the number of research studies published per year has been increasing. These fundamental research studies are necessary to advance therapeutics for Schaaf-Yang syndrome. So this is the 20-year journey from gene discovery to therapeutics, really from the year 2000 to 2020. So after the discovery of the Magell2 gene, several groups decided to create mice with a mutation in Magell2. These so-called gene-targeted mice, or Magell2 knockout mice, or simply Magell2 mice, were first described in 2007. The next step was to see whether the mice had any symptoms that were similar to what was seen in Prader-Willi syndrome, remembering that nobody knew about Schaaf-Yang syndrome at that time. Differences in physiology and behavior were found between Magell2 mice and so-called wild type or control mice. We know at that time that levels of Magell2 were tied to the 24-hour cycle. It was therefore exciting to see that levels of Magell2 were not only tied to the 24-hour cycle, but that the Magell2 mice actually had abnormal sleep rhythms. In fact, when maintained in constant light, they no longer slept in blocks of time, but were up running on a wheel at all hours. In mice, reinforcing the light-dark cycle was important for the mice to have blocks of sleep. In 2012, Dr. Schaaf demonstrated that mutations in Magell2 cause a neurodevelopmental delay that resembles but is distinct from Prader-Willi syndrome and what we know now is Schaaf-Yang syndrome. So a large series of case reports followed, demonstrating a wide range of body systems affected by Magell2 mutation. Over the past 10 years, preclinical studies in mice have shown that interventions can change outcomes in Magell2 mice. At the same time, Dr. Potts and others started developing a better understanding of the function that Magell2 functions inside the cell, how it performs in regulating the stability and the location of other proteins. Our focus was more on physiological defects in mice, and that was as one road to therapeutics. To test therapeutics, we first need to show that the mice have a difference from control mice that's large enough that we could see the effect of a treatment. In addition to the circadian rhythm and sleep disorders, the mice had excess fat mass and reduced lean mass, reduced endurance on a treadmill, spinal curvatures, specifically kyphosis, which is a top to bottom hump rather than a side-to-side curve that we see more typically in humans, reduced bone mineral density, and anxiety-like behavior. So this slide just illustrates one of the most consistent findings in Magell2 mice is that they have more fat and less muscle than control mice. On this slide and the next, the Magell2 mice are in red and the control mice are in black. You can see in this X-ray of a control mouse on the top and the Magell2 mouse on the bottom that the Magell2 mouse has much more fat, even though it weighs the same as the control mouse. Magell2 mice are also less active than control mice. So that's both measured by their spontaneous activity, as you can see on the left, and how much they run on a wheel that's placed in their cage on the right. So up till now, we've been looking at what happens when Magell2 is missing, but we also need to answer the where and the why of Magell2. So some proteins are active in every part of the body, and these tend to be proteins that make up either the structure of cells, or that make cells divide, or that make up the overall structure of organs. Unlike these widely expressed proteins, Magell2 is present in very specific tissues. In these cross sections of a mouse embryo, we see intense staining, so that's the dark purple staining, in the hypothalamus, which is the part of the brain right above the roof of the mouth. On the left, the dark purple spots show Magell2 expression at the base of the brain in the hypothalamus, but also in the cortex of the brain and in the tongue, which is a muscle. On the right, Magell2 is also moderately expressed in developing cartilage and in bone and in muscle. Many of the medical issues in Prader-Willi syndrome and in Schaaf-Yang syndrome can be traced back to this part of the brain, the hypothalamus. The hypothalamus is located deep in the brain and is a control center for many feedback functions that keep the body running smoothly. These functions include sleep, temperature, appetite, and regulation of hormones like growth hormone and reproductive hormones. The hypothalamus is very important to sense and respond to energy signals. And so it was interesting right away in the context of Prader-Willi syndrome because of the difficulties with appetite and obesity faced by people with that disorder. So fat mass is regulated by a hormone called leptin, which is made in fat cells. That hormone then travels to the hypothalamus where it tells the brain whether or not the body has sufficient fat reserves. So normally, if an animal loses weight, this feedback mechanism uses leptin to tell the brain to eat more and to become less active. This is the pathway that's broken in Prader-Willi syndrome, causing excessive appetite and weight gain. Now we know that leptin acts on both POMC neurons, those are the ones shown in yellow here, and NPY neurons. So we decided to look at POMC neurons specifically to find out whether individual neurons activate when we add leptin to them. On the graph on the left, you can see electrical activity in individual neurons in each of those colored traces. While the neurons in the control mouse activate with leptin, so at the spike where the green arrow is pointing, the neurons in the Magell2 mice don't activate, and that's shown by the lack of a spike where the red arrow is. This is likely the reason that the Magell2 mice gain excess fat mass, is a lack of activity in response to leptin in POMC neurons in the hypothalamus. So now that we've measured how mice carrying the Magell2 mutation differ from control litter mates, our next question is whether there are therapies that can normalize these differences. And specifically, can we bypass the leptin response defect using melanocortins? So are there therapies that can normalize these differences? And I'm going to talk to you now about some of the different preclinical work that we've done to look at therapeutics. Not only therapeutics that might address this defect in leptin signaling, but other types of potential therapeutics as well. So this was one of the studies that we performed looking specifically at melanocortins. So we see here the defect in the POMC neurons. POMC neurons release a substance called alpha MSH, and that's shown right where that yellow neuron is meeting the so-called second order neuron here in blue. Now what we can do is replace this alpha MSH, which is not being properly released by the POMC neurons, and replace it with a compound that mimics alpha MSH. And in this case, we used a compound made by Rhythm Therapeutics called setmelanotide. So we tested whether we could bypass that leptin response defect using a melanocortin that acts on the second order neurons downstream of the POMC neurons. And in this experiment, we injected mice with setmelanotide, this melanocortin that acts on second order neurons. In the top right-hand side, we see here that the Magell2 mice increased their energy expenditure when they were injected with setmelanotide compared to when they were injected with saline. And they also increased the number of horizontal beam breaks. So that's the amount of activity that they had when they were injected with setmelanotide. We can also see that their food intake was very reduced compared to when they were injected with saline. And so what that means is that even though the Magell2 mice have a defect in these POMC neurons and are no longer releasing alpha MSH, we can actually substitute a different compound for the lack of the release of MSH by POMC neurons. So far, we've been talking about these POMC neurons and how they respond to this leptin released from fat. Another type of neuron, these NPY neurons, also respond to leptin. These neurons are sensitive to a drug called diazoxide, which is already being used for other types of diseases. So on the right, we can see that feeding Magell2 mice diazoxide in their food for either 6 weeks or 12 weeks substantially reduced their body fat, although the Magell2 mice still had more body fat than the wild type mice. We didn't see any effect on lean mass with treatment with diazoxide, but diazoxide did reduce fat mass. Many of the studies on Magell2 mice were designed to look for therapeutics for Prader-Willi syndrome, and only by extension for Schaaf-Yang syndrome. Some, like diazoxide, oxytocin, setmelanotide, have already been tested in clinical trials in Prader-Willi syndrome. Although this table is difficult to read, it shows that survival and social behavior can be normalized by treatment with oxytocin. Food intake can be reduced by melanocortin treatment, as I just showed you, or by bariatric surgery, or with a cannabinoid inverse agonist, or with diazoxide, and that a fatty acid amide can improve bone mass. And these are all studies that were done in either the Magell2 mice that we've been using or Magell2 mice by Françoise Muscatelli's group in France. And so this table is just to show you that many, many preclinical studies have already been done with this mouse model of Schaaf-Yang syndrome, and that many of these discoveries are being translated into clinical trials. So far we've been talking about mice lacking Magell2, where in the body the gene is active, but not so much about the Magell2 protein. Dr. Potts is going to tell you more about the cell biology of Magell2, but I'm going to spend the last few minutes that I have talking about some of the work that we've been doing funded by FPWR to better understand what Magell2 does in the cell. So in general, MAGE proteins, which are here indicated in green, act in many different processes. They bind to and regulate the activity of different proteins, whose job it is to add and remove a very small protein called ubiquitin, which are these little orange balls here, from other proteins. Ubiquitin is a flag that targets these proteins called substrates, these blue proteins, targets them for degradation or to be sorted to a different location in the cell. Now, one thing we know about Magell2 is that it's a very large protein. It has a MAGE domain, shown here in green, and that's what makes it part of the MAGE family of proteins. It has a very unstructured front end. So the whole first half of the protein is unstructured, but a highly structured second half. And right here, this chart here in blue and red shows how disordered or unstructured the protein is. And you can see that the first half of the protein is very unstructured. And what that means is that it doesn't form a precise structure in the cell. It doesn't curl up into a predictable structure. So we know that some of the mutations that truncate the protein do so throughout the protein, but many of them truncate the protein right here at amino acid 666. And this is usually a frame shift mutation or a stop mutation that either stops the protein immediately or allows a very small little extra bit of protein to be made. So this truncation leaves only the first unstructured part of the protein intact. The MAGE domain, which is the working part of the protein, is no longer made. In the case of the common single base insertion and deletion, this extra abnormal protein may extend off the end of the Magell2 protein. And it's possible that this unstructured abnormal protein actually gums up cells. And we think this is one possible reason why the symptoms of Schaaf-Yang syndrome are in some ways worse than in Prader-Willi syndrome, where the whole Magell2 gene is missing, although that still remains to be seen. So one of the experiments that we've been doing is looking at a system where we could tag and then identify proteins in the cell that are close to Magell2. So in this experiment, we used either the entire Magell2 protein or only the second half that contains the MAGE homology domain. Consistent with other studies, we found proteins that are important for ubiquitination, like USP7, are close to Magell2. We also found a large cluster of proteins over here in red that are involved in RNA processing. And these proteins seem to bind only to the beginning of the Magell2 protein, which is the part that's still remaining in people with Schaaf-Yang syndrome when it's caused by a truncating mutation. Now we know that RNA is the intermediate messenger of the cell, and proteins that bind to RNA, this green structure here, regulate the location and the stability of that RNA. Each of the proteins marked here with the yellow star was found to be near Magell2 in cells. Overall, we found that Magell2 regulates the abundance of proteins that modify these messenger RNAs. Regulation of messenger RNAs is particularly important under conditions of cellular stress. And this suggests that the imbalance in the amount or the location of messenger RNAs could contribute to both Schaaf-Yang syndrome and Prader-Willi syndrome. So as a summary, cell and animal studies have helped us to understand how Magell2 regulates the abundance of many proteins that either bind to mRNA, which is the messenger of the cell, that regulate stress responses, that function in day-night circadian rhythm, proteins that sense the fat hormone leptin, and proteins that regulate the production of neuropeptides and hormones in the hypothalamus. And with these cell and animal studies, our hope is that we can test and develop new and known therapeutics that may be useful in both Prader-Willi syndrome and in Schaaf-Yang syndrome. So I'll stop there and take any questions that you might have. Thank you.
Thank you so much, Rachel. It was really interesting to see the list of compounds or hormones that have been tested in the Magell2 mouse. Our first question that came in was actually related to that list. You know, you mentioned diazoxide and oxytocin and this fatty acid amide. Are any of these being recommended off-label for people with Schaaf-Yang, or have these been limited only to models at this time?
That would be a question for Christian.
Is Christian still here? Maybe he'd like to weigh in on that. He may have stepped away. It was evening
Oh,
time.
I'm here. I'm here. I just had to unmute myself. So as far as I can tell, at this point, no systematic clinical trials of any therapeutic intervention have been done in Schaaf-Yang syndrome.
All right. So specifically diazoxide, it's generic, and at least in the US, it's widely available. Is that something that would be of interest to families? It's been tested in mice. It's been very safe for its indication use. Is there a reason why parents would want to try that in their Schaaf-Yang families or in their Schaaf-Yang loved ones outside of a clinical trial?
So I think this is a question we should ask a pediatric endocrinologist. So someone like Jennifer Miller from the University of Florida, who's been very engaged in Prader-Willi syndrome and clinical trials and endocrinology as it relates to PWS. I think this would be an excellent question for her.
Perfect. That sounds like a great idea.
I think as a whole, overall, the trials in Prader-Willi syndrome have really been focused on hyperphagia and on altering behavior around food. And so you have to remember that those trials are really focused on that. The animal studies are focused overall on physiology and a lot of different systems, but the endpoints of most of the clinical trials have been mostly around either hyperphagia, so that food drive, or around reducing fat mass. And if those aren't concerns in families, then those are not compounds that they would be wanting to try or even look at.
So have you
So I have an update here, sorry, because I knew that diazoxide came up once, and I looked back in my email trail with Jennifer Miller, who I just mentioned. In 2018, there was a family who contacted us about hyperinsulinemia in a child with Schaaf-Yang syndrome, where the endocrinologist wanted to consider diazoxide, and Jennifer supported that idea. And the family wrote back a few months later saying that they hadn't observed any hypoglycemia since the child was put on diazoxide.
And that's the indication for diazoxide, correct? It's intended to
treat hypoglycemia as a consequence of hyperinsulinism, yes.
Right. But the mouse study showed that it could improve body mass by decreasing body fat, correct?
Yeah, and it does also reduce blood glucose levels, which is that's its main purpose in medicine, is to be used with infants who have produced too much insulin.
Mm-hmm.
Yeah.
So on the flip side of the coin, how about lean mass? Is there any studies that have shown ways to improve the lean body mass?
So there's certainly clinical trials ongoing to improve, for example, lean mass in people with cancer cachexia, where they have reduced amounts of muscle, to improve lean mass in children with muscular dystrophy or in other disorders with muscle wasting. And at some point, it would be important to look at transferring some of that information over into the Schaaf-Yang and Prader-Willi communities to see whether there is any potential for improving lean mass in those children. But again, those studies tend to fall a little bit by the wayside because of the pressing need for treatment of hyperphagia in Prader-Willi syndrome.
So if hyperphagia is not the only potential endpoint for Schaaf-Yang syndrome, have there been any studies that have shown improvements in other areas that are relevant to Schaaf-Yang, such as cognition?
As Christian said, there are no studies in Schaaf-Yang syndrome, but of course, there are studies ongoing in autism spectrum disorder, looking at a number of different compounds, including oxytocin, And the intellectual disability community certainly is investigating a lot of different therapeutics. And the Prader-Willi syndrome organizations are looking as well at a lot of therapeutics for symptoms other than hyperphagia.
So many preclinical studies have been done in the mouse model, and you've mentioned that they're now being translated into clinical trials. Are any of these studies approaching human clinical trials for Schaaf-Yang phase 1 or phase 2?
So not that I'm aware of for Schaaf-Yang syndrome, but it's been an interesting evolution, because you have therapeutics that are already approved for use in humans that are now being used secondarily in Prader-Willi syndrome. Then there are brand new therapeutics that are being developed, and then of course they have to go through all of the animal studies, all of the cellular studies, and then all of the phase 1, 2, 3 clinical trials. And so there actually has been a lot of back and forth between animal models and clinical trials, where some compounds are tested first in mice, and some are tested first in humans and then tested afterwards in mice. And it's a very complicated process, because you really have to have a good reason for thinking that a positive result in an animal study will help approve for usually FDA approval of a therapeutic for use in humans. So the most straightforward therapeutics are the ones that have already been approved for other uses in humans, and those are the ones where it's easiest to go into large clinical trials, like diazoxide, for example. But unless Christian can correct me, but I don't know that there are any others that are going into clinical trials for Schaaf-Yang syndrome specifically.
Right. So given that this is going to be our last question before we turn over to Dr. Potts, given the rarity of Schaaf-Yang syndrome, is the current strategy to ride the coattails of the Prader-Willi group studies, and so that Schaaf-Yang can benefit from those? Or are we looking to try to get Schaaf-Yang specific studies so that there is an indication specific to Schaaf-Yang, or is the strategy somewhat a overlap of both of those?
So ultimately, the decisions about medical care are between the family and their physicians. In terms of clinical trials, I mean, anybody who's active in this community can influence how those clinical trials go. And we've seen that for sure in the Prader-Willi syndrome community, where FPWR is the organization that is fundamental to organizing clinical trials, because without the parent groups pushing for enrollment and advertising the studies, it would be very difficult to do those clinical trials at all. And Prader-Willi syndrome is a rare disorder, but it's not an ultra rare disorder. And that the pharmaceutical companies that are doing clinical trials rely very, very heavily on family groups to make sure that those trials can go ahead. But maybe Christian wants to comment on that as well.
So everything that Rachel said is obviously correct. I think there's great potential because there are multiple avenues for which we have evidence from preclinical studies related to the Magell2 gene and protein. The major challenge is that there are multiple challenges, but I think the major challenge is that the community is still very small. So in any given place, and in many cases even in any given country, there's a very small number of individuals that would be available for a clinical trial. And I personally believe that clinical trials using growth hormone and oxytocin are two of the first ones that should be considered. But even with that, you need to decide which one should you do first. You cannot do both using the same cohort. which then further decreases your ability, or it would have to be done by two differently entirely groups who both need interest in the condition and funding. So clinical trials are oftentimes, fairly expensive studies to do. But we are in touch with FPWR, with the patient community, the patient advisory board, and some of the other medical specialists and clinical trialists kind of thinking about clinical trials and planning those for the coming years.
Great. Well, Rachel, I would love to thank you for having joined us today, really having a great, robust conversation. Our next speaker is going to be Dr. Ryan Potts. He's joining us today from Amgen, and there he is.